ordered sequential reaction. So sequential means, let's say there are two substrates, A or B or S1, S2, then both have to bind in order to give you the products. And the sequence in which they bind may be fixed or may not be fixed. If it is fixed, then that is called ordered, or some other word like that, customary, or so it has to be in certain order. There are two substrates A and B, then A has to bind, then B has to bind. Whereas if it is, it doesn't matter, either A or B can bind in the first time, so then that will be random. So random, sequential, and ordered sequential means whether A binds and then B binds, or anyone can bind first. And then there is another type of reaction called the ping-pong reaction. So in a ping-pong reaction, both substrates don't have to bind. So substrate 1 binds, gives you the first product, then substrate 2 binds and gives you the second product. So it's like a game of ping pong ball, so that's why it's called that way. So these are the two types of bi-substrate reactions, which are designated in a diagram by drawing a horizontal line showing the form of the enzyme that happens at each step. So initially, when nothing is bound, you have the free enzyme, which is E. And then we are looking at this reaction, let's say S1 plus S2 being E1 plus E2, and it's catalyzed by the enzyme. So first, S1 binds, because we are looking at the ordered ones, which you show like with an arrow like this, pointing to the line, which means S1 binds, and when E binds to S1, it will of course become ES1. And then the second substrate must bind, so it gives you ES1, S2. And that is when the reaction can take place, which you show as an arrow like this. You may or may not have the arrow, but it is shown in parentheses. So ES1, S2 goes to E1, E2. So these are the two product forms, E1, E2. So this is a reaction that takes place, and then the products leave in an ordered manner. So P1 leaves first. So form of the enzyme that will be left will be EP2, and then P2 leaves, and you are back to the original form of the enzyme. So this is the order of events that happen, and when people derive kinetics or any other observation from that, it is based on this concept, which we are not going to go beyond this. In a random sequential, two have to bind, but doesn't matter what order. So if you have the enzyme here, so this is the random. And then E can bind to S1 or S2. So there are two possibilities. S1 can bind here, or another possibility that S2 can bind. So if S1 binds, you get ES1. This gives you ES2, and then the second one binds.
Oh, I've seen this before. I know this is.
And this will then react, convert to the product. And once the product is formed, again the products are released in a random order. So that means you can release let's say P1 first, then you are back to P2, P2, or if P2 is released first, then you get P1, and then the next one is released, and you get back to the original enzyme. So this one will be P1 in this case, and this will be P2. So the order in which the substrates and products bind can be anyone, so that is a random sequential order. And then you have the ping pong kinetics, which is actually more common. So in this, you have the enzyme form that binds to the first substrate. And then the first product leaves. So when the first substrate binds, it gives you ES1. And that itself reacts. The enzyme converts S1 to P1. So it will go to S1 to P1. And then the P1 leaves. So first product leaves. So S1 came in, P1 left. They are not the same molecule. That means the remaining part of this will also be not the same as the starting enzyme. So this is a modified form of the enzyme that stays behind. In other words, some functional group from S1 stays attached to the enzyme, and the remaining part of the molecule leaves as the product P1. So the enzyme is in a modified form, and then the next substrate comes and binds. Sorry, S2. So you get E star S2, and this again reacts to give you EP2, and then the second product leaves, and you are back to starting enzyme. So as a catalyst, you regain your enzyme at the end of the reaction. So in general, these are the three types of bi-substrate reaction. What prevents S2 from binding first? If it is an ordered one, I said S1 has to bind first, but it's a movement of molecules. They are all moving in all directions and they hit each other and they bind. So if S1 can bind, why doesn't S2 bind in the beginning? So for all these enzymes, this was explained by Koshland. We already talked about this. Koshland's induced fit model. Remember that explains the specificity of the substrate for the substrate. So when the substrate binds, there is some binding site for the substrate. But then the enzyme undergoes a conformational change and it binds to it more tightly. So the groups come closer to the substrate and binding it tightly and doesn't allow something else to get in. So before the first substrate binds, the active site, the binding site for S2 is not even created. So the enzyme doesn't have a clear binding site for S2 that is formed only after S1 binds. So after S1 binds, the enzyme undergoes a conformational change, and in that new active site, there is a site for S2 to bind, and that's why S2 always binds later. It's not just a random thing that S1. If S2 goes in first, then what happens? S2 won't go in first because there is no binding site that is created after S1 binds. As I said, the ping pong mechanism is more common for the bi-substrate reactions. To give one example, if you take glucose, glucose is abbreviated as GLC, and you react with ATP. That gives you ADP plus glucose 6-phosphate. We will study these reactions later in the carbohydrate chapter, but just to introduce these numbers and letters here. The 6 is the position of the ring. So when you glucose forms a ring, the carbons are numbered 1 through 6. So this is the carbon 6 of glucose. And the P is a short form for phosphate. So sometimes the P is written with a circle to tell you that this phosphate has 4 oxygens also. It's not just a phosphorus, but phosphorus with 4 oxygens. And biochemists are lazy, they don't want to write the oxygen, so they just write a P. So glucose 6 phosphate. So this reaction follows a ping pong mechanism, and in fact, any reaction that uses energy from ATP follows this mechanism. So it is not just about glucose. Anywhere you find ATP as providing a phosphate to something, this is how it happens. It happens in this type of a mechanism. So what is the first substrate? It is ATP always binds first, so enzyme binds to ATP. And so of course you get enzyme ATP complex, and then that gives you enzyme attached to phosphate. Bound to ATP. So this is the reaction. So the ATP, the name says there are 3 phosphates, and the terminal phosphate, also called the gamma phosphate. So alpha, beta, gamma, these are the 3 positions for the phosphate. The gamma phosphate is transferred to one of the amino acids of the enzyme, and you have ADP as the product, so that product leaves. So ADP is your first product, and you are left with enzyme bound to a phosphate. Phosphate is attached to the enzyme. And then the next substrate comes in, which in this case is glucose. So you get glucose bound to enzyme phosphate. And then this phosphate is transferred to the glucose. So glucose gets the phosphate, and you get your free enzyme back. So the glucose phosphate leaves, and you get back the free enzyme. And this is understandable because as you will see throughout all biochemical reactions, whenever we study metabolic pathways, everything is about energy. You understand that any reactions require energy. So if you want to form a glucose phosphate bond, that bond requires energy. Whenever you are forming a bond, it requires energy. And that energy is coming from the ATP. So here you have adenosine attached to ribose, attached to 3 phosphates. And once again, I'm not writing the 4 oxygens with each phosphate. So there is a bond energy between the 2 phosphates. These bonds have energy between the phosphates. And that is the energy that is used to form a bond between glucose and phosphate. So how do you transfer the energy? You transfer the energy by not hydrolyzing any bond. If you just break this bond, then phosphate comes out. That is of no use. One phosphate comes out as inorganic phosphate ion. You cannot bind it to an enzyme, to any substrate, because you don't have the energy. Energy is lost. So this phosphate is never really simply hydrolyzed. This is transferred to something. That means enzyme, remember, have many amino acids with the OH group, for example, serine. So it may be transferred to a serine or to histidine, or something of threonine that has a OH group. So you are preserving that energy of this bond into the enzyme phosphate bond. And that energy is then used to form a new bond between glucose and the phosphate. So that's why this is a common mechanism for all enzymes that require energy. You cannot waste the energy by simply breaking that bond. You have to transfer the phosphate to something, which is the enzyme. That's why it's a catalyst. And then the enzyme transfers it to the second substrate. sometimes the energy is already in the substrate itself, and so you don't need it. For example, if you take glucose, fructose, this compound is sucrose, let's say a common sugar. And then you can react, there's another substrate which is a free phosphate, and that will give you glucose phosphate plus fructose. Here also you are forming glucose phosphate, you are forming a bond between glucose and phosphate, and you are not using ATP. Why? Because there is energy in this bond. The glucose and fructose bond has energy, so you don't need another source of energy from ATP. And this will also be a ping pong mechanism, so if you want to do this, it will first give you enzyme attached to phosphate, and the first product is fructose that leaves, and then this reacts with the glucose to transfer the phosphate to glucose. So once again, the energy is conserved. The energy in this bond is conserved in the energy. Sorry, this is we are not using glucose. Second substrate is phosphate, and this one is glucose. So this bond is broken, fructose leaves, this part of the substrate remains attached to the enzyme, and then the second substrate comes and you transfer the phosphate to the glucose. So in this case, the energy in this bond will be still conserved in the glucose enzyme complex, and then the phosphate reacts with that. So you can form a bond to free phosphate without ATP, provided your substrate has energy there. So this is also a ping pong mechanism. All these enzymes, I'm not naming the enzymes, but you can always figure out what the enzyme will be. So this is adding a phosphate on sucrose. So this is called sucrose phosphorylase. This one we will study in glycolysis chapter. That is the first reaction of glycolysis, and this enzyme is called hexokinase. There are two types of enzymes that we will frequently see related to phosphate. One is a kinase, the other is a phosphatase. Kinase is an enzyme that adds a phosphate to a certain molecule, and phosphatase are enzymes that remove a phosphate. And these are very common reactions. So this is hexokinase because it is adding a phosphate to glucose. And that is the first reaction of glycolysis, which we will study later. All right, so one last thing from this part of the chapter, and then we will move on to enzyme mechanism. So this is at the junction of mechanism and how the enzymes, enzyme kinetics, you can say. And that is another type of inhibitor. Called a suicide inhibitor. And some people don't like the word suicide, so they call it a mechanism-based inhibitor.
The other one.
Inhibitors.
Okay.
Now I mentioned that as pharmacists, most of the drugs you will be dealing with, or if you are into research, that your goal of synthesizing new drugs will be synthesizing new competitive inhibitors of some enzyme responsible for some disease. But then there is a problem with the competitive inhibitor. The name competition says that they are in competition with the substrate. So how much of the competitive inhibitor do you need to inactivate the enzyme? It cannot be a 1-to-1 ratio because it is always competing with the substrate also binds there. So in order to inhibit one molecule of enzyme, you need many molecules of the inhibitor. And that makes all the competitive inhibitors actually much weaker inhibitors than the suicide inhibitor, which works on a stoichiometric basis. That means one molecule of enzyme will need one molecule of the inhibitor. So this is your dream drug for the pharmacist. When they are doing research, this is what they want to discover. This is what they want to synthesize. It may not always happen, but this is what you want to do most of the time. So what is a suicide inhibitor? As the other name suggests, it's a mechanism-based inhibitor. So these inhibitors bind to the substrate just like a competitive inhibitor because it looks like the substrate. But in fact, it looks so much like the substrate that the enzyme cannot even differentiate that this is not the substrate. So it starts the reaction. In organic chemistry, you studied mechanism and you know it's not just one step. There are multiple steps in a mechanism. So same way with enzyme reactions also, there are multiple steps in the mechanism. And the enzyme cannot differentiate between the inhibitor and the substrate because they look so much alike. So it starts the mechanism, goes through some steps, and then it stops because the inhibitor you have designed in such a way that the next steps cannot happen. So here is your inhibitor. The enzyme, if it was a competitive inhibitor, then it will bind to the active site and then come off. All it does is blocks the active site and prevents the substrate from binding. But this suicide inhibitor binds to the enzyme and starts the reaction. So that means, let's say you have step 1, step 2, step 3, like that. There are several steps in this mechanism itself. And in one of those steps, the enzyme is covalently bound to the intermediate. So here is an enzyme that is covalently bound to the intermediate in some form of the intermediate. And the next step requires that, let's say, a hydrogen has to be removed. So there is a hydrogen here and there is a base at the active site, and that will remove that hydrogen. So this is, I'm just generalizing a reaction, but as you know, every inhibitor works in a certain enzyme for a certain reaction, so it will all depend on what reaction it is. So let's just make up a reaction here. In this reaction, there is a hydrogen that needs to be removed by one of the basic amino acids. And then it will give you the products. And as the clever scientist, you have designed this inhibitor, which looks exactly like the substrate, except that you have put a fluorine instead of the hydrogen. Fluorine is a very commonly substituent for hydrogen when you want to develop a inhibitor. Why? Because fluorine and hydrogen are the closest in size. Hydrogen is very small, and what is the atom that is most close to that size? That is fluorine. Because fluorine is in the second period of the periodic table. As you go towards the right, size decreases. So fluorine is the smallest atom that is very close to size of hydrogen. But there is a big difference between hydrogen and fluorine, and that is in the electronegativity. If something removes a hydrogen, that is taken out as a H+, right? So we can say that this H+ ion is removed by the base. Fluorine is so electronegative that you can never remove a F+. This reaction cannot happen. So the enzyme has gone through the first 1 or 2 steps, and then it stops because it cannot remove that fluorine, because you have put that fluorine in place of a hydrogen. And now you are stuck, or the enzyme is stuck, bound to this inhibitor. It cannot go back, it cannot go forward, and the enzyme is permanently inactivated. So this is a mechanism-based inactivation of the enzyme. Competitive inhibitors do not inactivate the enzyme, they inhibit the enzyme. That means when the inhibitor comes off, it is active again. Whereas in this case, this is covalently bound to the inhibitor and stops the reaction because the enzyme goes out of circulation. So enzymes are catalysts, same catalyst repeats the same reaction thousands of times or millions of times, removing one molecule of the enzyme will slow down the reaction a lot. And you're using 1-to-1 ratio of the inhibitor to the enzyme. So this is your dream drug that you want to make to have high activity at a very low amount. Some examples of this, one example in which I mentioned this fluorine and hydrogen, you'll find that in anti-cancer drug. That's how the anti-cancer drug works. It is a 5-fluorouracil that is used. And we will talk about that in the last chapter or so. Another example of a mechanism-based inhibitor which wasn't designed. So obviously you can think that all these mechanism-based inhibitors are designed. They are synthesized in the lab, and that's how you think about the mechanism first, and then you discover or make the inhibitor. One inhibitor that was already made, already found in nature, and it also works by this mechanism is penicillin. So penicillin is an antibiotic, it's a natural antibiotic found from fungus. But when people understood the mechanism, they found that this is also a suicide inhibitor or a mechanism-based inhibitor because the penicillin remains permanently bound to the enzyme. And how does it work? And everyone knows penicillin is also called a, yes?
Oh, cell wall synthesis?
It inhibits cell wall synthesis, yes.
Beta-lactam.
How does it do that? It inhibits cell wall synthesis. We are not going into the details of that, but exactly how does the inhibitor work is that it has a beta-lactam ring. Right? Everyone knows that, and penicillin is also called a beta-lactam. And what is a beta-lactam? We studied that in organic chemistry. What is a lactam? Lactam is a cyclic amide, like a lactone is a cyclic ester. So if you have a carboxylic acid and a OH in the same molecule, then they can form an ester and form a ring. Or if you have a carboxylic acid and an amine in the same molecule and they form an amide linkage, then it will form a ring. So what size ring it will form depends on how many carbons are in between the carboxyl group and the amine. So accordingly, you will get a 4-membered ring, 5-membered ring, 6-membered ring. So if it is a 4-membered ring, that is called the beta-lactam because the amino group is at the beta position from the carboxyl group. So if you have a carboxyl group and alpha, beta, and amino group. So if this forms an amide linkage with this one, the ring you get is a 4-membered ring. So this is a beta-lactam. This is part of penicillin. So the rest of the molecules are not drawing. So this is penicillin. And it works because of this beta-lactam ring, which are very unstable, because it's a 4-membered ring, so that's why it is unstable, so it breaks open easily. The way the cell wall synthesis works, as someone mentioned, this inhibits the cell wall synthesis of bacteria. One of the important reaction in that is to have a peptide, the end of which is a D-alanine. So these are the last 2 amino acids in a cell wall peptide chain. So here is your chain, and this is one group in that, and there are numerous such groups on that chain called the peptidoglycan. And in this reaction, if you have another chain that also has the same thing, so this one is, or there's actually a lysine here, so this links to here and this one comes out. So when the cell wall is made, it makes these numerous chains, 1 chain, 2 chain, and 3 chain. Each of them have this, and between 2 chains, they are cross-linked one amino acid forms a peptide bond with another amino acid, and this last peptide bond breaks. Once again, energy in that last peptide bond is used to form a new peptide bond between the chains, and that's how you cross-links the cell wall and makes it strong. And that is needed for the bacteria to survive. If you prevent that cross-linking, then the cell wall will be weak. And to do that cross-linking, there is an enzyme that does that. Of course, all reactions are catalyzed by enzyme, and the substrate for that is the D-alanine bond. That is the substrate. It is attached to the rest of it, but this is the main reaction site where the reaction takes place. And if you draw the structure of D-alanine and D-alanine, it looks like a beta-lactam, but without the ring. So you can draw D-alanine without the four-membered ring, but the structure is similar, and that's why penicillin binds to that site. Yes?
Can you explain that again?
Which one?
The alanine.
So if you have the bacterial cell wall, so they will have many chains of carbohydrates, and in those there will be a peptide ending with alanine. Right, it's actually D-alanine. This is an exception to the rule, as I mentioned. All amino acids are L. Bacteria make these a D, and there is a reason why they make it a D. Whenever bacteria puts a D amino acid, it is to trick the host so that the host proteases don't break the peptide. So bacteria have evolved to be more clever than us. So if they want to infect the host, they need to have a protection, because otherwise the host proteases will break it down. So that's why they put an unusual amino acid. In fact, all these peptide bonds, there are several amino acids here, they're all very unusual ones. So the D-alanine, the bacteria will make a stronger cell wall by cross-linking this. So this will be cross-linked to this one, this will be cross-linked to this one. So they are cross-linked in different positions, and that makes the cell wall strong. So in order to cross-link, this is what the bacteria does. One peptide linkage here consisting of 7 amino acids. So one of these will form a bond to the other one. So you need to form a peptide bond between this one. Lysine, remember, has the extra amino group. To form a peptide bond, you need an amino group. And that's why it has to be a lysine. It cannot be any other amino acid because they don't have the amino group. So you have to have an amino group, extra one, and that can form a peptide bond with the D-alanine. And the energy required for that is in this last peptide bond that leaves. So it is essential that the D-alanine bond breaks so that it can form a cross-link structure, and the penicillin inhibits this enzyme that breaks this bond or that combines the lysine with the D-alanine. So the substrate for that enzyme, which is called a peptidyl transferase. So this is this bond, or this last two amino acids is the substrate, and the enzyme, of course, recognizes that and binds to that. And if you draw the structure of that, that looks like a penicillin, but without a ring. So what happens? Yes.
What's right on the bottom?
Another peptide chain with a lysine followed by D-alanine and another D-alanine. Just like this one. This also has a lysine. I'm not writing those. There's a lysine here, there is a alanine here. All the chains have the same sequence. So this lysine combines with this D-alanine, and that's how they are cross-linked. So when the penicillin binds to it, it looks like the D-alanine, but it also breaks the bond, but then this is still attached to the ring. So if, let's say you break this bond here, but then this rest of the molecule is still attached to this because it's a ring. You have to break two bonds to completely separate them. Whenever you have a ring, if you break one bond, the rest of the molecule is still attached to the enzyme. So in this case, when D-alanine bond breaks, this leaves. In this case, when you break the bond, then the rest of the molecule is still attached to the enzyme. And that's how it inhibits the enzyme. It inhibits in a 1-to-1 ratio because when penicillin inhibits the enzyme, the reaction actually starts because the substrates look very similar. The enzyme cannot differentiate between them. It starts, goes to the first step, and then it stops because the rest of the molecule hasn't left. In this case, the G-alanine leaves, and that's why the enzyme finishes the reaction. Here, this doesn't leave because this is still a ring. You have to break 2 bonds to separate the rest of the molecule. And that's how this works. So this is another example of mechanism-based inhibitor or suicide inhibitor, and there are many other examples, of course, and they all work on this same principle, that the enzyme goes through a couple of steps of the mechanism and then it cannot go further because you have designed the substrate that way that next step cannot happen. And because it has already gone through some of the steps, it cannot go back also, and that's how the enzyme is permanently inhibited, and that's why it's called the suicide inhibitor, because it goes through some of the steps and then it kills itself. So that brings us to enzyme mechanism. How do enzymes speed up a reaction? So rate of reaction, as you have studied in chemistry, is any reaction. Remember, enzymes don't change the thermodynamics of the reaction. They only change the rate, and so rate will be dependent on the rate constant times the substrate. It doesn't depend on the products. If you have 2 substrates, then it will be K, S1, S2. So K is the rate constant. And K is also given by another equation which also you have studied in general chemistry. So K is equal to A e to the power minus EA over RT. Brings back memories from general chemistry? No? It should.
Not good ones, hopefully not much. Terrible memory.
All right, that means the rate of a reaction depends on these. And if you are doing a reaction in a test tube without enzymes, as in inorganic chemistry, what do you change to speed the reaction? If you look at this equation here, everything is a constant. This is the energy of activation. Energy of activation, also called the activation energy, and A is called the Arrhenius constant. So only thing you can change is the temperature. So that's why whenever you do a reaction in the lab, you increase the temperature so that your reaction happens faster. You don't have that liberty in the cell temperature is also fixed. So how do you then increase the rate of reaction, which that's what the enzymes do. And enzymes do that by increasing this or by decreasing this. So these are the 2 mechanisms by which enzymes function. They increase the rate of reaction by increasing this constant called the Arrhenius constant, or by decreasing this, because remember this is the negative sign here. So negative means it's an inverse, so if you lower energy of activation, it gives you faster reaction. So enzymes work by lowering the energy of activation or increasing the constant here. And what is this constant dependent on? It depends on the frequency of collisions. So that constant tells you how frequently the molecules collide. And it's not just collision, it is also productive collision. So this is the frequency of productive collisions. So two molecules first of all have to collide to react, and not only that, they have to collide in the right orientation. So if you are doing a reaction between a hydroxyl group and a carboxyl group, and the molecule is a large one, so here is the molecule, and there is this group here X that reacts with this one that has Y. So these two ends must come together to react. If this end and this end collide, then that's not a productive collision. So the rate of reaction depends on the number of productive collisions, and enzymes increase that. So let's understand then what are the mechanisms by which enzyme works, and the only way they will work is by either increasing this or decreasing the energy of activation. Alright, so I'm going to erase that. So here is the active site where the substrate binds, and let's say there are two substrates, whatever mechanism that we saw before, any one of those. So this one has to bind here, and then the enzyme waits for the second substrate to come, and then it gives you the product. If you are doing without a catalyst, then S1 and S2 have to come together. There is that frequency of collision is very low. As you know, in the gas phase or in solution, frequency of collision is very low. How do you increase that frequency of collision? If enzyme binds to, so this will have a low frequency, but it is bound here, and then this doesn't leave. In a random collision, they are always moving. Even after colliding, they are moving. So the enzyme actually binds to the substrate and waits for the S1, S2 to come in. And that increases the rate of collision because the first one is already stuck there, it's not leaving, it is in one place. If it did leave as in a random movement, then the frequency of collision will be lower. And also it increases the productive collision because if the substrate has this shape, the active site has this shape, then the enzyme, the first substrate will also be binding to it in this manner only. It goes into the active site in one direction only. And if the reaction is supposed to be with a, let's say, glucose or phosphate, and that happens to be here, that will always be pointing at the active site in one direction only because that's how the active site is designed. And if the phosphate needs to be transferred to the enzyme, that will happen because this end will never get in here. And that means the frequency of productive collisions increases because of the substrate specificity of the active site. Because if there are two substrates, they will bind in exactly the same orientation because it has to interact with the amino acids at the active site, and that it can do only in one orientation. So this is the this is one of the mechanisms by which enzymes increase rate, that is the proximity. Proximity and orientation, which means it brings the substrate together in the right orientation. So that increases the rate of reaction. Another thing that increases, so these are increasing the value of heat. Looking at how it increases the value of heat. Another mechanism by which it does that is with the metal ion. Many enzymes have metal ions at the active site, and metal ions are positively charged. So if the enzyme, for example, binds to ATP, let's say, so here is your enzyme, and at the active site of the enzyme there will be a magnesium ion. Usually most enzymes that use ATP as one of the substrates either also require magnesium or calcium, which is bound to the active site, which is of course positively charged, and ATP, as you know, is adenosine with 3-phosphate. So the phosphates have negative charge, all the phosphates have negative charge, and the magnesium helps to bring that to the active site. So the positive charge of the metal ion attracts the negative charge of the substrate, and that's how it increases the rate. It's not just a random collision, but it is an interaction between the substrate that increases the rate. And by the same concept, it doesn't have to be a metal ion. It can be, in general, any electrostatic interaction. So instead of a magnesium, it can be, for example, a carboxylate ion from the aspartate or glutamate. Sorry, so that will be negative zero. It can be a lysine. So lysine will have an amino group, or arginine will have amino group, not exactly like this, but nitrogen with a plus, and they will attract negatively charged substrate. So if this enzyme catalyzes reaction with a negatively charged substrate, there will be a reactive site, a lysine or an arginine somewhere there that is attracting the substrate to the site, and that increases the frequency of the collision. Or if you have a positively charged substrate, then there is probably a aspartate or a glutamate with a negative charge on them that attracts the positive charge of the substrate. So the right amino acid is at the right position in the active site to attract the substrate towards itself, and that increases the frequency of collision, and also it has to be in the right orientation because of the substrate specificity as well as the charge. For example, ATP, the negative charge is at the end, and that is the phosphate that is transferred, so that automatically goes into the active site in the right orientation. So that brings us to another concept here. In order to be the right charge, as I said, lysine must be positively charged, aspartate must be negatively charged. Yes?
What does that say about the...
Aspartate and glutamate, they have negative charges. Lysine, arginine, they have positive charges, and also you can have histidine that, as I mentioned, can be positive, negative, both, depending on the circumstances. So when will they become charged? At a certain pH, right? As we have seen before, the pH, if the pH is below pK, it is protonated. If it is above pK, it is deprotonated. So if there is an enzyme that has an amino acid that is essential for its activity, how do you figure out that? How do you tell that this enzyme has a certain amino acid that is responsible for the activity? You can do that by measuring the activity at different pH values. So we take the enzyme and measure its activity at various pH. And let's say you find this one. You get a curve like this at a certain pH. So let's say this is 4. So below 4 it is inactive, above 4 it is active. So this is you did the experiment, you got this curve. Now what do you conclude from this? What amino acid at the active site is important for its activity? Something that gets deprotonated. Remember, below pK it is protonated, above pK it is deprotonated. And right here it is deprotonated. So that means, for example, a carboxylate ion from aspartate, and here it will be COOH below the pK. So you would conclude from this that this enzyme has an important amino acid at the active site which must be in the deprotonated state to be active. If it is protonated, that means it has a proton, then this will not be active. I wrote here 4, but if this happens at let's say 9, then what would you conclude? If this is, you get the same graph, but this happens at this is at pH 9. Then you would say that this is a NH3 plus, and this is NH2. So it loses the proton and becomes active. Usually it doesn't happen like this, nothing works at pH 9 or above. But if it did, then this is what you conclude. That the enzyme has an active site of the enzyme has something, the amino acid, that must be deprotonated in order to be active. So usually it doesn't go to 9, as you know, all the enzymes work maybe between 4 to 7 or 8, that range. So if you get a graph like this, and let's say this is happening at pH of 6, then what will you conclude? You will conclude that the enzyme is active when this is in the protonated form, and it is not active when it is in the deprotonated form. And from the change, the pH at the change, where the change takes place, you can guess which amino acid it is. If you look in the pK table and say this must be an aspartate or a glutamate, because that's where they have their pK, and what can be this one that changes the activity at around pH 6. So that, as I mentioned, the only amino acid that has a pK near neutral is histidine. So you would say that there is probably a histidine that is important for the activity, and it must be in the protonated state for it to be active. If you get a graph like this, if this is your pH activity curve, then what do you conclude then? So there are two graphs combined. So this is happening at pH 4, and let's say this is happening at pH 6. So you would say that there are two amino acids at the active site, and one has to be deprotonated, the other has to be protonated. So this one has to be in the deprotonated form, and this amino acid must be in the protonated form for it to be active. And as I said, from the pH where the change takes place, you can guess what amino acid it is, but it will be a wild guess, and sometimes you may go wrong. And that's what happened with this enzyme. So this is the activity curve of the protease pathway. And at pH 4, you may think that this is an aspartate because of glutamate. And at pH 6, you may think this is a histidine. But that is only a guess, because as I said before, all the pKa in the table that you have in your book, they are for free amino acid in solution. That is not meant for the enzyme. The active site of the enzyme doesn't have much water in it. So the whole concept of pH doesn't even apply at the active site. It's a totally different concept that pH plus pOH must be 14. All those apply when you have lots of water around. And the active site doesn't. So sometimes you may be wrong, and in this case people were wrong to assume that this is aspartate and this is histidine. It turns out that this one is actually histidine, and this one is a cysteine. The 6 maybe a different number. I'm just making up these 3 to 7. So just by going by the number and looking in the table, you cannot tell exactly that this must be the amino acid. Sometimes you may be right, sometimes you may not be right, because the environment at the active site is different from the free solution. So you have to do other experiments to find out that this is histidine, which doesn't have a pK at 4, it has a pK of 6, but histidine and this environment at the active site has this pK, and histidine must be in the deprotonated form, and cysteine must be in the protonated form to be active. Alright, so these are for increasing the value of A. You can also increase the rate of reaction by decreasing the value of energy of activation. And energy of activation is the energy needed. So this is the reaction. This is the progress of the reaction versus energy. So this is the energy of the reactant, product, transition state. Right? That's how in general chemistry you learn all reactions happen. So the energy of activation is this energy. Molecules must have this energy to go to the products, and then the reaction will happen. So enzymes work by decreasing the energy of activation, and there are two ways you can do that. How do you decrease energy of activation? You can make the substrate unstable, or you can make the transition state more stable. Both ways you can decrease energy of activation. Raise the energy here, or bring this energy down. So you have to have a method for stabilizing the transition state. That's one of the mechanisms by which some enzymes work. Or you have a method of increasing the energy of this, but you cannot really increase the energy of a molecule. So one way to do that is to make an intermediate. And that's how the enzyme works. So that mechanism is called covalent catalysis. Covalent catalysis, it forms a covalent bond to make an intermediate that has a higher energy. So instead of having this energy diagram, it will now have an energy diagram like this. So here is your enzyme, it gives you an intermediate, and that gives you. So instead of going all the way from here to here, you're going in 2 steps, and this is now your energy of activation. There are 2 steps, and the slower step will be the rate determining step that decides the rate of reaction. So the enzyme is breaking up the reaction into 2 parts so that the intermediate is unstable compared to the original reactant, and so it is closer to the transition state, and that gives you a faster reaction. And we already saw that reaction. Many enzymes work by this mechanism. They will make an intermediate that is unstable. For example, if you take glucose plus ATP and put them in a test tube and come back the next day to see how much glucose phosphate you made, you find you made 0 amount. Alright, so ATP is a source of energy, it has energy in it. So you expect the phosphate to transfer to glucose, but it won't. The reaction has a very high activation energy. The molecules don't have the energy to climb this much activation energy to go to the product. So this reaction never happens. Even if you heat it 100 degrees and leave it overnight, it will still not transfer the phosphate to the glucose. So it will not give you glucose phosphate. And that's where you need the enzyme. So what is the enzyme doing? So with enzyme, you are forming this unstable intermediate, remember, which is the enzyme phosphate complex. So here is enzyme plus glucose plus ATP. In fact, it is, as we saw before, it is a ping pong mechanism, so the first step is only ATP, and then glucose comes into the picture. So first it forms an enzyme phosphate intermediate, which has a higher energy than the enzyme itself or the ATP itself. So enzyme phosphate is unstable, and that's why it has a higher energy, and that brings it closer to the energy of the transition state, and so it can go to the glucose phosphate. So this plus glucose gives you glucose phosphate because you are closer in energy, and that speeds up the reaction. So this is called covalent catalysis, and many enzymes work by this mechanism. That means you form an intermediate that is more unstable, and that is closer to the transition state. So all ping pong mechanisms will also follow this, and the purpose of having that mechanism is to have an intermediate that is more unstable, and that's why you get the reaction faster. And how did people prove that? What will be the proof of this mechanism? Proof will be to isolate this and show that this exists. that is unstable, but if you have a mechanism of isolating that and or prove that it exists, then you have proved the mechanism. And people have, and so people who are actually doing pure research on enzyme mechanism like this, if they can isolate an intermediate, that is again their dream for proving a mechanism, that I have isolated this mechanism, this intermediate, and that proves this mechanism. And so enzyme hexokinase that has to phosphate, that has been isolated, and people know where the phosphate binds, which amino acid binds to that. So that's how you study these mechanisms. So this was over the. Another one can be acid base catalysis, and as you have studied many reactions in organic chemistry that are catalyzed by acid or base. For example, you take a protein and you react it with 6 molar hydrochloric acid, heat it for 4 or 5 hours, you are going to break the peptide bond. So you are hydrolyzing the peptide bond catalyzed by an acid. So hydrolysis, remember once again, the peptide bonds are formed by removing a molecule of water between 2 amino acids. So if you want to break that peptide bond, you have to add water to get your carboxyl and amino group, so that reaction is a hydrolysis reaction. And that is the reaction that happens in the process of digestion. So you can do that in the lab without an enzyme, but it will be a very difficult reaction. As I said, you need 6 molar HCl and heat it for hours, 100 degrees or more, and then you get the reaction. Or if you want to do a base catalyzed reaction, that also you can do, but that also requires the same high concentration of sodium hydroxide at high temperature for a long time. So both are difficult reactions, but the enzymes of course do that at body temperature at a much faster rate. So let's look at the hydrolysis of the peptide bond. So here is a peptide bond with the rest of the protein here, and you are going to break this peptide bond without an enzyme. So that means you are adding water. So how does that happen? So let's say you are doing in presence of an acid catalyzed reaction. So this will react with the acid, so that gives you NH2. And then this bond will break. How does it break? With the help of a base. So a base reacts here, and this bond breaks. So that's the base acid catalyzed reaction. Acid reacts with the amino group, and then a base breaks that bond, and you have separated the 2 parts of the peptide bond. Now, what base is that? If you are doing a reaction in presence of an acid, as I said, 6 molar acid, what is the base you can have in the same solution? You cannot have a strong acid and a strong base in the same solution. So whenever you are doing a reaction in a strong acid, you will look in the mechanism and see that there is also a base involved in the next step. And that base can be only water. In presence of a strong acid, the only base you can have in aqueous solution is water. So this base is a water molecule. So that's the base that you can have. You cannot have a strong acid and a strong base at the same time. Or if you are doing the reaction in presence of a base or base catalyzed reaction, then the base reacts here. So let's say you have a strong base hydroxide ion that reacts here, and then this will react with an acid, which is water. So the base reacts at the carboxyl group, and the amino group picks up a proton from water. So in presence of a strong base, the only acid you can have is water. In presence of a strong acid, the only base you can have is water. But this is how acid-base catalyzed reactions work, and you separate the peptide bond. If you are doing this reaction with an enzyme, like all the proteases, you cannot have strong acid or strong base, but the mechanism is still the same. There is an acid and there is a base. And what are those acids and bases? They are the amino acids at the active site of the enzyme. And there are numerous examples. I'm just showing with a peptide bond, but there are numerous examples in biochemistry where an acid or base is required for the catalytic mechanism, and that acid or base has to be one of the amino acids at the active site. You cannot have a strong acid or a strong base. So if you are doing at the active site, so here is your active site of the enzyme, and here is your peptide bond. So there has to be an acid that binds to the amino group, and there has to be a base that binds to the carboxyl. Right? So let's say this is the mechanism, the acid binds to the amino group, and there must be a base that binds to the carboxyl group, and they together give you the reaction. So what kind of acid will you have? You can have a lysine with NH3 plus, or arginine with NH3 plus. That is the acid that is present in the active site, so that reacts with the peptide bond or amino group of the peptide bond. And you also need a base, so the base can be a carboxyl group of aspartate. So that will react here. So the right amino acid at the right position of the active site will act as an acid or a base. Yes?
So how would you access as an exam question?
We are not doing an exam. You can make any question. You can make it right now. Let's make a question. If you understand it, don't worry about the questions. All the questions are based on your understanding.
I'm not sure what is understanding, so if it's an NH3, it's supposed to bind to an acid, and if it's a
No, NH3 is the acid, carboxylate is the base.
so what is
This is how the enzyme provides the acid and the base. You cannot have a strong acid or a strong base in aqueous solution. That acid has to come from somewhere. And the enzyme has an amino acid at the right position to give that reaction. That amino acid is not a strong amino acid, a strong acid or base. But the reason you have a faster reaction with a stronger acid or base, or a more concentrated reaction, is again the frequency of collision. As you know, between two reactions without an enzyme, the rate increases if you increase the concentration of the reactants. At the active site, the concentration is already very high because everything is concentrated in one place. Nothing is moving. So the concentration is very high, so you don't need a strong acid or a strong base. If you have anything that works as an acid, that works the same way as a concentrated acid or a strong acid, because it is at the right position on that site, closest to the substrate. If this is your substrate peptide bond, and this is the lysine that is just staring at its face, it is the closest amino acid it can have, and its activity will be like that of a strong acid because of proximity. It is in the right place, and the enzyme has been designed. Nature has designed the enzyme so that when the substrate binds, this lysine will be at the right place to provide that proton. Or this carboxylate will be at the right place, very close to the carboxyl group, and so it can act as a base, and you don't need a strong base. In fact, even if you need, you are never going to get that in aqueous solution and pH 7. So this is the reaction that can happen without an enzyme, which takes a long time, high temperature, but the enzyme does that under milder conditions because it always has the right amino acid at the right position to function as an acid and base. So any reaction that is acid or base catalyzed and the enzyme catalyzes that will be catalyzed by some amino acid at the active site that is providing the acid or the base. So that is another, and I'm just giving examples here like with the peptide bond, but there are numerous examples. Any reaction you pick is catalyzed by enzyme. Any biochemical reaction is catalyzed by enzyme that increases the rate of reaction, and it has to follow one of these methods by which it is. All the enzymes will work by the proximity and orientation. That is true for all enzymes. Some enzymes work by the covalent catalysis method, some work by the acid-base catalysis method. So these are the various methods by which enzyme activity increases, or rate of reaction increases. As I said, you can also lower the energy of activation if you lower the energy of the transition state. Till now we have been looking at ways of increasing the energy of the reactant, but you can also decrease the energy of the transition state. That will give a faster reaction. So some enzymes work by that mechanism, and that is preferential binding. So the enzyme binds to the transition state better than it binds to the substrate. And that's how it lowers the energy of the transition state. So once again, as I said, as a pharmacist, your goal is to design competitive inhibitors that look like the substrate. But then you are thinking always about the mechanism in which the substrate binds to the active site and then it gives you the product. So you want something to look like the substrate. But then there are some enzymes that don't work that way. They increase the rate of reaction because they bind to the transition state better than to the substrate. And so if you want to design a drug that inhibits that enzyme, you have to make something that looks like the transition state so that you can inhibit the enzyme that way. For example, if you have proline, let's say proline racemase that will convert D-proline to L-proline. So you would expect that you will have, it's a 5-membered ring, as you know. So here is a carboxyl group and an amino group. So proline is the only amino acid that has a secondary amine. And how will you make it a D or L? If it is L, how do you make it a D? Or if it is this is the alpha carbon, and if you want to if there has to be alpha carbon between the carboxyl group. So if you want to make a D or L, you have to remove a hydrogen and make a carbanion. So there is a hydrogen here. If you remove that, you get a carbanion, which is a planar structure, and then the enzyme will put another hydrogen from the other side. So if this hydrogen came, let's say, from the front, then the next hydrogen comes from the back. So where does the hydrogen go and where does the hydrogen come from? There must be an amino acid at the right position, at the right place in the active site that is removing a proton, and then another one that is adding a proton back. So it is always done that way. There must be an amino acid that is doing it. One amino acid will be on one side of the protein, the other will be on the other side. So one removes a proton this way, and another adds a proton from the other side. And so that will give you the, if you have started with the L, then this will give you the G. So that's how all rasterizers work. You remove a hydrogen from one side, add a hydrogen from the other side, then you have changed the configuration. So the point is that this goes through this transition state. So this is your transition state in which a proton has been removed and a new proton is being added from this side. So from this proton is from some amino acid, which we don't know. But some amino acid remove this proton and another amino acid is adding this proton. But the point here is that the intermediate is a carbon ion which is a planar transition state. And this enzyme is known to bind to the transition state stronger than to the substrate, which is the D-alanine or the L-alanine. Proline, D or L-proline. So, if you are the person trying to design some drug to inhibit this enzyme, what will the drug look like? If you design something that looks like proline, that doesn't work because the enzyme recognizes the transition state better than the substrate. So you want to design something that is a planar structure. Here it is a planar structure. And what would you use to have a planar structure? You can use a aromatic ring that is planar. And it was found that if you want to inhibit this, some structure like this one that has a double bond here, I don't remember what form they use, but something that is a planar structure, which is aromatic, and that will inhibit the enzyme. Alright, so that is another concept. This is the modern trend for pharmaceutical companies to discover drugs that inhibit a certain enzyme because the enzyme binds to the transition state better than to the substrate. Alright, so these are the 6 different mechanisms by which the enzymes function. Yes.
So in the transition state, what's written on the top right?
Top right. Transition state is formed by removing a proton. So remember, D and L will differ on whether the amino group is on the left or right. So one proton is removed, and then a proton is added from the other side. So if you have changed the direction of the proton, then you have changed the orientation. So that's what the enzyme does. One amino acid removes a proton, another amino acid adds a proton. Yes.
Is the transitional state the one with the extra charge?
Yes, this one has a charge. On the substrate, yes. Because the hydrogen has been removed. So all these structures that I'm drawing, as you recognize, they are not complete structures. I'm just drawing the important part of the molecule. So when it's the proline, you know what the proline should look like. You can find that. So as I said, some enzymes will follow one mechanism, another enzyme follows another mechanism, some enzymes follow more of these mechanisms. Serine proteases are ones that actually follow almost all of these mechanisms. So what is a serine protease? Serine protease is not an enzyme that recognizes serine and breaks a bond. So you may be misled into that understanding, but that's not what it means. A serine protease is an enzyme that breaks a peptide bond based on a serine at the active site that is essential for the reaction. So the serine at the active site participates in the catalytic mechanism. And these are called serine proteases. The examples you already know, these are the digestive enzymes, trypsin, chymotrypsin, elastase. So these are digestive enzymes secreted into the small intestine that breaks peptide bonds. From your biology class, you know that all these digestive enzymes are made in an inactive form, because otherwise they will digest the organ that is making them. So they are all made in an inactive form and then they are converted to the active form. Anyone? Yes? That's called elastase. You may not have heard of it. But these are the peptide bonds that cleave after they recognize the lysine. Lysine and arginine. Every enzyme has a specificity. So trypsin recognizes a lysine and arginine on the peptide and cleaves after that. Wherever there is a lysine or an arginine, it will cleave after that amino acid. In the same way, chymotrypsin cleaves after aromatic amino acid, for example, tryptophan, phenylalanine, tyrosine. So wherever there is a tryptophan, phenylalanine or tyrosine, the chymotrypsin will leave that peptide bond. So that's how digestion takes place. You have a long protein. Wherever these amino acids are, the enzyme will cut there. And then elastase cleaves after alanine. So it recognizes alanine and after the alanine. So all these enzymes are actually made in the form of a zymogen. Zymogens are inactive form of these enzymes. So zymogen example will be trypsinogen. That is converted to trypsin after it comes to the small intestine and then it digests peptide bonds. In the same way, you have chymotrypsinogen. That gives chymotrypsin, and the zymogen of elastase is proelastase. So these are all cleaved to give you the active peptide, active protease that will then digest the food. How do these work? All these enzymes work because of an active serine. So that's what we are studying here. This is just the background from your biology. If you haven't studied biology, you should know these basic facts. Not important for this class. But what is important is the mechanism of the serine protease. Alright, so serine protease, as the name suggests, has a serine at the active site that participates in the catalytic mechanism. And this is how it works. So at the active site, of a serine protease, there will of course be a serine. And then there will be a histidine somewhere here. And then there will be an aspartate. So these are the three amino acids that are in the active site of the serine protease that are responsible for the catalytic activity of these enzymes. Now these amino acids are not one after another in the primary sequence. As you know, protein folds, and so you have a protein that folds like this and forms the active site like this. So these three amino acids happen to be close to each other at the active site, but they are not next to each other in the primary sequence. To know what is the actual position in the primary sequence, each of the amino acid comes with a number. For any protein, every amino acid has a number. So if you look at the numbers in your textbook, there will be numbers like one is 57, the other is 75, the other is 192. So they are far apart in the primary sequence, but they happen to be close to each other in the three-dimensional structure of the protease. Alright, so in order to break a peptide bond, as we saw before, you need an acid or a base. For example, we saw you need a hydroxide to catalyze the reaction for peptide bond breakage, because hydroxide is a strong base. In organic chemistry, you learn that you can use a stronger base than hydroxide. That will give you a better reaction. And what will be a stronger base than hydroxide? Hydronium is an acid. Stronger base than hydroxide is an alkoxide. Like from methanol, you get methoxide. From ethanol, you get ethoxide. So those are stronger bases than hydroxide. So this is methoxide, also written as MEO minus or ETO minus. So these are stronger bases than hydroxide. So if serine has a OH, of course the OH is on a carbon of serine. Serine, remember, has the formula. So this is your serine. If you can remove the proton from here, that is like an alkoxide ion, and that will be a strong base. And that is how all these serine proteases work. They remove a proton from serine to make this strong base, which then catalyzes the hydrolysis of peptide bonds. Now in water, how can you have a strong base like this? Usually the hydroxide has a pK above 15, and so you cannot really make them. Making them in the lab itself is difficult, and maintaining them or working with them is difficult because they are too reactive. So how does the cell make an alkoxide ion to catalyze the reaction? And that it does because of these three amino acids positioned in the right place. And these are called the catalytic triad. So the catalytic triad are these three amino acids next to each other in the three-dimension at the active site, but not next to each other in the primary sequence. So trypsin will have that, amitripsin will have that, all these serine proteases have that. And as I said, every amino acid has a number, and these numbers will not be the same for the different enzymes. Every enzyme has a different sequence. So if you look at what number of these amino acids, they will be different for each of these enzymes. But the fact is that these three come together, close together at the active site, and that helps this serine lose a proton and become a strong base. And once you form that strong base, that will hydrolyze the peptide bond, just like we saw before. So all serine proteases work this way. So you have a aspartate that has an extra carboxyl group, and somewhere very close to that is the histidine, which has a 5-membered ring. So there is a nitrogen. Histidine has a 5-membered ring with 2 nitrogens. One of them has a H, the other doesn't have a H. That is histidine. If you look at the structure of histidine, that's how it will look. There are double bonds there, which I'm not drawing complete structures. So this is aspartate, this is histidine, and then there is a serine with a OH. So these happen to be close to each other within distance that is sufficient for forming hydrogen bonds. Remember, hydrogen bonds need a certain distance, and The aspartate can form a hydrogen bond. So if I write the bond with the hydrogen like this, the hydrogen from the histidine goes to the aspartate because you form a hydrogen bond, so the hydrogen is somewhere in between. And in the same way, this nitrogen will form a hydrogen bond to this OH, and that's how this bond to the serine breaks. So the hydrogen is removed from serine through this chain of amino acids to the aspartate. So the hydrogen of serine basically is going to aspartate, not directly, but through the histidine. So this type of system is called the catalytic triad, or it is also called the charge relay system. So serine proteases work by the charge relay system, where the charge of aspartate is now on the serine, because the hydrogen has moved from the serine to the aspartate, not directly but through the histidine. So histidine gets this hydrogen and this hydrogen goes to the aspartate. So the idea here is then to have the serine with the O-, which is a very strong base. It may not be a full charge, but even if it's a partial charge on it, because the hydrogen is still attached to it. So this serine then will break the peptide bond. So the peptide bond so this is your peptide bond, the serine oxygen reacts here and this bond breaks. And that's how you form a bond . So the amino, one half of the peptide bond is now broken, and the other half is still attached to the serine. And then this bond needs to break, and that will be broken by an aspartate. So you can this can get a proton from another aspartate, and then the bond breaks, so you get the same. So another amino acid helps to break this bond. So it will be a water molecule also that because it's at the end, it is water that is hydrolyzing the bond. So the idea is to break the bond between the carbon and the nitrogen with a strong base, which is formed because of the catalytic triad. And this is the common mechanism in all of these serine proteases. So if they all work by this mechanism, then why do they have a specificity? The mechanism is the same. How can they have different specificity? For example, trypsin and chymotrypsin and elastase, they have a different amino acid specificity. And that is because the catalytic reaction happens in a different place than the binding of the substrate. So there is a binding pocket of the enzyme where the lysine or arginine binds in trypsin. So here is the binding site where the lysine binds. Lysine, as you know, has a couple of carbons followed by NH3+. And then the reaction actually happens here. So this is the protein that is being cut. And lysine binds to this pocket, and the catalytic triad is somewhere here. So serine, aspartate, and serine, histidine, and aspartate. So the catalytic triad is in a different place than the binding pocket. So the reaction will happen only if lysine binds to this pocket. And so the shape of this binding site resembles the shape of the lysine in depth. And not only that, there will be a negatively charged amino acid, like a carboxylate from an aspartate, that will bind to the amino group of the lysine, and that's how it recognizes the lysine or arginine. In the same way, if you look at the binding pocket for chymotrypsin, chymotrypsin will have an aromatic ring here. So you expect that these amino acids will be hydrophobic. So the aromatic amino acid binds there because of the hydrophobic amino acids at the binding pocket. And the length of the binding pocket is sufficient for serine, for phenylalanine or tyrosine. So it is the right length and the right hydrophobicity of the amino acid that determines the specificity of chymotrypsin. So this is trypsin. And another one I mentioned is elastase that recognizes alanine, and alanine has only has a CH3 as the side chain, so that's a very small one, and so elastase then will have a binding site like this. So we have a small binding site with hydrophobic amino acids here, and that will bind to the CH3 of alanine. So they all have the serine and histidine and aspartate, but the binding pocket is different, and that's how they have these specificities. Yes?
Sorry, I still don't understand what a binding pocket is.
All these amino acid has a certain specificity for the amino acid. As I mentioned, trypsin will cut after lysine and arginine, and chymotrypsin cuts after tyrosine, phenylalanine, and tryptophan. Aromatic ones. So how does the enzyme recognize these substrates? And they all work by the same mechanism for the catalytic reaction. So besides the catalytic reaction, besides the catalytic triad that exists in all of them, there is also a region where the lysine will bind for trypsin. Only if lysine is there, then it will bind there, and then the reaction will happen. If it's a different amino acid, that will not bind into that pocket. So the specificity happens because of the existence of a separate binding pocket at the active site besides the three catalytic amino acids. Yes?
When the serine turns into the alkoxide ion, does it break off from the rest of the enzyme?
No, it's still there, part
It
of it.
stays on?
Because it's not a full alkoxide ion. The hydrogen is basically shuttling between the serine and the histidine and the aspartate. So it may not be a full charge on that oxygen. Yes?
Is the last one elastase?
Elastase. So in the same way, there are other enzymes that work with other active amino acids at the active site. For example, there is another type of protease called the aspartate proteases. For example, all the other enzymes that digest protein. Anyone knows what other enzymes digest protein? From your biology class. Proteins are digested in the stomach and the small intestine. So in the stomach there are the enzymes pepsin and renin that digest protein, and then it goes to the small intestine where these enzymes will digest the rest of it. So the stomach pH is low, and there are two enzymes, pepsin and renin, that digest proteins, and these are different types of proteins, and they are called aspartate proteases. So these have two aspartic acids at the active site, which acts as the acid. So aspartic acid, one will be protonated, the other is not, and so some of them don't have two, some of them will have only one. So there's an important aspartic acid at the active site that makes it as an acid or as a base. So these are acid-base catalyzed reaction also, where the proton is coming from in COOH at the active site. So these are called the aspartic bases. All right, so that's the last thing on this chapter on mechanism. The next thing that we will be doing will be the coenzymes and cofactors, enzymes, and then we will move on to metabolic pathways. So we will do that. All right, so stop here. Any questions?